Hi there, it's Lauren Whitehurst, a TA for Bot 2800C, Plants and Human Affairs. Today I want to show you a little bit about Lab 2, where we looked at many different essential oils. So all around me smells wonderful. We have lots of lovely essential oils, and these of course are plant extracts for the most part, serving different purposes for the plants themselves. They could be anti-herbivory, something like that. But luckily for us, some of these oils have medicinal properties. So today what we're going to do is called a Kirby-Bauer assay, where we are going to test some essential oils to see exactly what sort of properties they have and how strong they are. So essentially what we have created are plates that have a bacterial lawn, and they're divided into 4 sections. These 4 sections correspond to these 4 different numbers that we have here. They are listed in your lab manual as so. The first one is water, the second one is soap, third one is an oil, essential oil, and the fourth is a second essential oil. So these are actually small dishes filled with these liquids. For my first lab, my oil A was tea tree oil. My oil B was birch oil. Alright, so I want to tell you how to do this experiment. First, you must obtain one of these plates. They're actually pre-labeled, which is really nice. So if you kind of want to keep that lid on as much as you can to avoid contamination. But we do have a bacterial lawn here, okay? Now, one of the first things we want to do is sterilize. Every quadrant has already been labeled, but we want to sterilize our forceps and grab a cotton circle, and these are essentially filter paper. Dip it in a treatment of liquid, place it within that quadrant, and repeat until you're finished. So let's go ahead and get started with that aspect of it. So here we have our forceps. We have a loose paper disc. We have here a lamp that will allow us to easily sterilize our forceps over and over. We also have 70% ethanol, which we have here sub-aliquoted into a smaller beaker. Okay, then let's grab our lighter, so friendly big lighter, and we're gonna light this flame here. So of course we're gonna move it away from the ethanol stations. Ethanol is everywhere. Here we go. So let us light that, and now we have a beautiful flame. All right, great. Let's flame our forceps. First you want to dip them in ethanol. Okay. You can let the excess run off. You want to pass these ethanol drenched forceps away from the ethanol but through the flame. It's called flaming your forceps. It's sterilizing them. That's probably a bit too long, so they should be sterile unless I touch something else. Okay. So ideally, let's just cap this so we don't have an open flame going. So we're going to deprive it of oxygen. Boom. Flames out. I'm going to grab the lid off, keeping very carefully. Trying not to desterilize my forceps. So I'm going to keep mine in this direction so I know, even though I took off the lid, what corresponds to 1, 2, 3, and 4. 1 is water. So, water, as you imagine, on a bacterial lawn won't do much. So this can be used as a control to make sure that the experiment itself is working. So water compared to soap, these two different controls will show you two different things. So let's place very gently our first disc. This is also known as a disc assay, so if you've heard of it referred to as that, that's not wrong. Okay, so we have the first quadrant filled. I'm going to pause for a second while I sterilize. Alright, great. So we're ready to grab our second disc. There we are. Make sure to grab just one. They do stick to each other. So, and you can either lay it in there and pull it back out, but you just don't want it to be dripping wet. Okay. Placement for number 2, our soap. So since this is a bacterial lawn, this disc itself, disc 2, should have a bacterial inhibition zone around it. So after it incubates for a week, we're going to check these again. There should be an area around this where the soap itself has eaten or destroyed the bacteria necessarily. So let's sterilize again, 1 second. Okay, disc number 3, sterile, forceps. This is tea tree oil, so I'm actually going to just let that set in there. Pull it out. There's a very thin layer of oil on the bottom of these. Tea tree oil is a wonderful oil. It's very versatile. It has many different properties. Okay. Sterilization break. The final disc for our assay, in this case, in my lab sections we used birch oil. So let's carefully transfer our last piece. Okay. All right, so that is how you do the Kirby Bauer assay. And let's turn off our flame. So I'm going to put my lid back on. Remember, always when you're working with petri dishes and things like that, you want to avoid as much contamination as possible and things entering your petri dish. So after you're done with that, we have this special tape here. So you need to secure the lid to the bottom with this tape. And also, you're going to want to make sure you put your initials on there so you know which one is yours. And in a week, so we're going to let these incubate and then we will get those results. All right? Thank you for your time and have a good day.